In this unit, we're going to be looking at nutritional genomics, and this part of the lecture is just going to provide an overview of some of the concepts related to nutritional genomics. With this, I'm expecting that you've already reviewed some of the basic genetic information that you've had in the past, so things like the structure of chromosomes, DNA, genes, gene replication, transcription, translation, all of those types of concepts. Make sure that you've reviewed that as you move forward. So in this, we'll look at kind of specifically some of the issues, genetic and genomic issues as they relate to nutrition. So first of all, there's a couple of basic definitions that I wanted to go over. These are kind of my basic definitions, I guess. There are some more formal ones out there, but there's often a differentiation between nutrigenetics and nutrigenomics. A lot of what we'll focus on really is more nutrigenomics, which is often also interchangeably used with nutritional genomics, but there is a little bit of difference between nutrigenetics and nutrigenomics. So nutrigenetics is how genes affect the way dietary components are processed. And I use the word dietary components because that really could be a variety of things, whether it's a specific nutrient, whether it's a dietary pattern, whether it's a dietary supplement, any of those types of things. So essentially when we're looking at nutrigenetics, we're looking at the gene structure and how someone's gene composition affects the way those components are processed or metabolized. When we look at nutrigenomics, we're looking at how the nutrients or bioactive compounds impact gene expression. So I'm going to put this in a little bit more of a graphic visual representation as well that I think kind of helps illustrate this difference a little bit. Again, these differences to some extent are I don't know if they're not arbitrary, but they can, a lot of it relates to similar things, but just to kind of clear this a little bit. So essentially on this screen, we kind of have, if we think about it with nutrigenetics and nutrigenomics, we can almost sort of divide right down the middle. And on the right side over here, we have what we would think about as nutrigenetics. So one example I have listed here, and again, we're looking at the genes, how the gene is in that individual person affecting how some type of nutrient or food compound is metabolized. And so for one example, and some of you may have heard of this congenital disorder, it's called sucrase isomaltase deficiency. So this is a genetic disorder that's interesting in Alaska because it actually has far higher prevalence in Alaska and Greenland and some of that those northern areas, far higher prevalence than other populations. So it's an interesting disorder to look at. But essentially, sucrase also isomaltase deficiency is this genetic disorder where the sucrase isomaltase gene has a mutation that causes the enzyme to be malformed. So remember, enzymes are proteins. We've already looked at that. And so when the gene that codes for that protein has some sort of variation or mutation in it, it can impact the way that protein, in this case the enzyme, is formed. And so then in this case, what that impact is that the person with that genetic mutation, which therefore causes the protein not to perform how it should, then that person is not able to break down sucrose or maltose because that's what that enzyme does. When we looked at enzymes and talked about enzymes, we really talked about their critical importance in nutrition, and this is an example of that. So essentially, this is an example of nutrigenetics because the person has a specific gene mutation that causes them to not be able to metabolize or break down a particular nutrient, in this case, sucrose or maltose. So if the person omits those from their diet, there's no evidence of disease. They can live perfectly healthy lives. However, if they eat those foods with those compounds, then they'll have symptoms because they aren't able to digest them and metabolize them. So that's kind of an example of nutrigenetics. On the other side of the screen, I have a nutrigenomics example. So in nutrigenomics, what we're looking at more so is really an interplay between the gene makeup, so maybe a little difference in the gene or a variant in the gene, and some type of nutrient. So an example that I have listed here is the ApoA2 gene, for example, and we'll talk in a minute about exactly what that means in terms of the terminology. What does ApoA2 CC variant mean? But just kind of for now, for the concept of thinking about what nutritional genomics is, basically, if someone has this variant of the gene and they consume a high-fat diet, they're more, and specifically high saturated fat, they're more likely to develop obesity than someone without the gene variant who also consumes that very same diet. So in this case, the nutrient, so saturated fat, is actually interacting with how the gene is functioning. But that interaction can be somewhat predicted by the gene variant. So in this case, the person doesn't necessarily need to completely omit a particular nutrient, but it is going to have some interaction with the gene that then will affect other health outcomes. So let's look at what these gene variants mean, or how we kind of describe this, what I have here with the ApoA2 CC variant. What does that mean? So a gene variant can really be anything that causes the gene to be different than what is most common in the population. And I sort of am hesitant to use the word normal in a population because some variants are actually fairly common. So I wouldn't consider it to be abnormal to have the variant, but it's less common than the other type. Sometimes the type that is more common is called the wild type. Um, there's different terminology, but essentially there's one variation of a gene that's most common typically among the population, although that's not always true. Sometimes there's several variations and a couple are equally common. But for the purposes of kind of understanding this, thinking that there's a version or allele of a gene that's more common. So then when we look at variants, it's something that's a little bit different. So what that variation can be is different. So I have some types of gene variants listed on the screen here: insertion, deletion, replication, copy number variations, and single nucleotide polymorphisms, or SNPs. So we'll look at a little bit about what each of those are. Another little bit about terminology is we often call it variance when it has some, perhaps makes some difference in outcome, but we really typically, the term mutation is reserved for things that have more severe outcomes when that gene is altered. So often the term variant is used when it's a slight difference that may impact phenotype, or what the actual person looks like, or how their physiological function is impacted, or that sort of thing. But mutation is a little bit more severe typically. The picture on the screen kind of shows the really rough layout of a gene. So we'll talk a lot about genes. One thing, always when you look at these kind of schematics, they're very simplified to help think about it in a simplified way. But remember, it is much more complex than this really is. Another thing to remember when we talk about gene variants is remember that each person who has a normal genome has 2 copies of every gene, because they get one set of chromosomes from mom, one set of chromosomes from dad. So they're going to have 2 copies. So when we look at gene variants, they can have the variant, so the different gene in one of those chromosomes, or a different version of the gene in one of those chromosomes, or in both of them. So when we look at terminology and things, remember that everybody has 2 copies, and so they could have the variant in one or the other. So each gene's basic structure has a promoter region, a coding region, and then a termination sequence that kind of ends the gene. Within the coding region, there's lots of different details in here. So this is just very basic, but just to kind of help you think about, if we're talking about an insertion, for example, that could be an insertion of another part of a different gene. It could be a replication is similar in that maybe part of the gene is replicated, or there's another copy of that part of the gene, and then put put in there. Copy number variations are similar. Sometimes there's a certain part of the gene has a certain number of copies of the same sequence within that gene, and so there may be more or less than the normal amount of those copies, and that can have implications as well. There can be a complete deletion of part or a large section or small section of a gene, so maybe this part or something is just completely missing from the gene. So those are a variety of the types of variations that are referred to or studied or looked at, and then really looking at how that impacts protein, um, uh, making proteins, and then the function of the body based on how those proteins were made. So the last one that I have on here is the one we'll actually focus on quite a lot, and that's a single nucleotide polymorphism. So a single nucleotide polymorphism is when just one nucleotide is different or replaced, so the polymorphism there. So I'll look a little bit more detail at that because that's where a lot of research, where there's a lot of research and work towards clinical application of nutritional genomic information. A lot of that is based on SNPs. So we'll look a little bit more specifically at that. I like this image to illustrate what SNPs are, but I also like it because it helps us think about this really in the context of how a gene works. So I'll talk about SNPs here, but I'm also going to kind of sidebar into some of the basics about genes to just kind of refresh your memory on that and help you think about it in the sort of the larger context. So remember that each gene is a relatively small piece of DNA. However, it's still a really long sequence of base pairs. So in this image, you see three bases that are one small part of a particular gene. So on the left, we have, and this is related to sickle cell anemia is the example, but it could be many other parts of a gene. So this right here is the three bases that are a small part of the sickle cell gene. So the gene involved in sickle cell anemia is actually the HBB gene, which stands for hemoglobin subunit beta. So this gene is found on chromosome 11, and it's actually 1,600 base pairs long. So we're looking at three little pieces here, but this is just, again, a small piece of the really large gene. So I only point that out just to remind you that when we're looking at SNPs, we're only considering one very small piece of a given gene. Sometimes there's actually multiple SNPs on the same gene that can impact function, and sometimes those SNPs interact with each other. So there can be lots going on. We're just really looking at a tiny piece. So remember that three nucleotides, again, just kind of reminding terminology and that sort of thing. So like the three nucleotides that I have marked here, those three nucleotides comprise a codon. And then different codons code for different amino acids. So that's kind of how the message gets through. And remember, in the process of making the protein, DNA gets copied over to mRNA or messenger RNA, which is what then actually codes for the protein. Another thing, just kind of a reminder, is that in RNA, there's one different nucleotide than DNA. So in RNA, the uracil or U replaces the thymine in DNA. So normally in DNA, A is going to pair with T, but when it becomes RNA, that is uracil instead of thymine in RNA. So just kind of a reminder there. So in this case, when we talk about kind of SNPs and thinking about what SNPs are, let me erase my what's there now. So when we talk about SNPs, we see in this one that the codon, the CTT, is found in normal hemoglobin. So this might be referred to as the wild type. And we can see actually that it's this T right here, that when we look at the mutated hemoglobin or the variant of this gene, instead of a T, we have an A there. So that's the single nucleotide polymorphism, the substitution of one single nucleotide within that codon. So in this case, what happens when that nucleotide is replaced with a different nucleotide, instead of making one specific amino acid that we need, so the glutamine, instead we make valine, because remember, these codons are specific to one amino acid. So this then makes a different amino acid in the full protein structure of hemoglobin. So again, to kind of put it in context, we're talking about one amino acid, but hemoglobin is actually made up of a lot of amino acids. It has 4 polypeptide chains, and each of those chains are made up of either 141 or 146 amino acids. So we're talking about one small piece of a large protein, but in this case, it has very severe implications. In some cases, a single nucleotide polymorphism or a SNP, one little place where it's different, doesn't make much difference at all, or makes a very small difference. So in this case, it's significant, but it doesn't always have to be. So there's a lot of research, as I mentioned, in clinical application in nutritional genomics as they relate to SNPs. So it's important that you really understand what these are and how to interpret genetic and genomic tests that utilize SNPs, so that you understand what they're getting at and just what that means, and being able to explain that to someone. One other piece to remember here is that we have 2 copies of DNA, and so 2 copies of each chromosome and each gene. In this case, in this schematic, we're just looking at 1 copy. So I point that out because someone could have 2 copies of the mutation or the variant, or they could have 2 copies of the normal version or the wild type, or they could have 1 of each. So if someone has both copies the same, that would be called homozygous, so the same. So they could be homozygous for the normal or wild type, or they could be homozygous for the mutated or variant. Or they could be heterozygous, which means that they have 1 copy of each. It varies a little bit depending on what the variation is, but typically if someone has 2 copies of the variant, the outcome is going to be more severe, which kind of makes sense than if they only have 1 copy of the variant, but not always true. And sometimes there's multiple different variants that can happen, and some have more severe outcomes than others. But just again, to keep in mind that those are the options, to either have 2 copies of the normal, 2 copies of the variant, or 1 copy of each. Because genetic testing has become relatively inexpensive, there are a range of genetic tests available either direct to consumer or through a healthcare provider. I don't expect that you know the details of what each of those mean for nutrition recommendations, but I do want you to know how to read an example of a test and explain what the lab who is doing the test is providing in terms of that information that they put out. So we'll walk through a sample in a minute, but first I want to touch on a few general areas related to nutrigenomic testing. First of all, let's talk about ethics. Ethics is a huge concern in really all areas of genetics. So if you're working in that area, you really need to explore that a little bit further than we will in this class. But one basic ethical consideration is whether there's enough research to have the genetic test really be able to change clinical recommendations. So that's an issue because we think about if someone pays the money to get a genetic test and they get information that may or may not really impact their care or make any difference in what they do for health, is that a good thing to do? Is that worth the money spent? Those kinds of things. There also sometimes can be genetic information that can be bothersome to some people, worrisome, cause unnecessary worry, those types of things. So lots of issues there. But for this class, and what I want you to really think about when you think about this area, that there's a lot of research in this area. Some of it is pretty solid, some of it's pretty weak, some of it's really mixed. So it's just important that you are understanding what is and what can and can't be said based on the information we have and the research we have, and that you're able to communicate both the strengths and the limitations of the existing research to consumers, clients, the public, whoever it is you're working with. So really, again, if you're educating people in this area of what the meaning of their genetic tests are as it relates to nutrition interventions, you need to really understand what research is out there on that. The other area to understand are some of the typical methods for testing. So very often these tests look at some variant of the DNA, most always that I'm familiar with. Very commonly they look at SNPs, but sometimes they may be looking at an insertion or deletion or something along those lines. But you want to be familiar with what those different variants are that they're looking at. Again, I mentioned this before, but be aware that sometimes SNPs interact with each other. So just because someone has one particular SNP, they may or may not respond the same way to a dietary intervention or some other type of intervention, depending on whether or not they have another particular SNP. So it gets a little bit complicated, and I think that's being looked at more and more, but it makes it sometimes difficult to know the best way to interpret a test. Even if we have the genetic information, what do we do with that? So let's shift a minute, and I want to take one example of a nutrigenetic test that is available. An example, I have the website here where I pulled a PDF from that shows a sample report from a company called Nutrigenomics. I'm using this as an example, not to promote it or to not promote it or anything like that, but simply because they had a nice example online of a test, and I just want to highlight some key elements of it that you will find if you search other companies as well. Other sample test results have similar types of things. So I want you to start to be familiar with what type of information someone might get if they did one of these tests, and so you could explain that to them, what that means. So again, this is a 23-page PDF document that I just pulled from the website I have listed there. And so I'm just going to highlight a few pieces of it that I think might be helpful in learning how to sort of apply this nutrigenomic information. So this is for someone who, an example of someone who sent in for a test, and I can't remember for sure. Typically what they will do is have people do a buccal swab, which is just a little swab in their mouth. Remember, because we are looking at just at DNA, we can get that from most any cell. So cells in the mouth are very easy to get. So that's a common way to send it in, but it could also be done with blood. It could be done with other cell types, as long as the cell can handle whatever, if it's being mailed in or whatever. So that's kind of what someone would send in, and then this report would come back because they're just doing analysis of the actual DNA itself. So this report gives a variety of sample of a variety of nutrients that interact with gene variants. So again, I'm not using this as a way to say something's right, wrong, indifferent, but just how you might interpret this type of a report and help a client or the public understand it. So in this case, they have some dietary components that they're focusing on, and you can see those listed here. This shows you what genes they are looking at to test for gene variants. So with each one of these, there's a specific gene, and then they're looking for the risk variant or something that might put someone at a higher risk or lower risk. So you can see there are some genes they're looking at deletions, and then the ones that have these, that lets you know they're looking at SNPs. So they're looking at whether they have the C or the T version and how many copies of that they have, and how that equates to risk based on the literature that they're using. So this is kind of a summary, and I'm just going to highlight a couple of these to show you how the more detailed report would look on here. Sorry, get out of my little writing tool so I can get down to the next page. Okay, this one is looking at vitamin C. And so again, just pieces of this report that if someone brought this to you and said, hey, I just did this testing, tell me what it means. So the GSTT1 is the gene that they are looking at. And so this gene produces a specific protein, the glutathione S-transferase enzyme. So again, pretty typical in nutrition that we're looking at the production of enzyme proteins, because we know that's going to impact how something is processed and broken down. Something that gets sometimes confusing, just popped into my mind, that I will point out with genetics and genomics, is that often the gene and the protein have very similar names. So you have to be a little bit careful about that when you're reading, just to kind of point that out. In this case, they are looking at either an insertion or a deletion. So the insertion that they're looking at is considered functional, or would be the normal or wild type that we talked about, while the deletion is where someone would have an issue with that. So then they aren't producing the glutathione S transferase enzyme sufficiently, or typically it's sufficiently. Sometimes, often it's not that they don't produce it at all, but it's not produced sufficiently or as well. So this is telling that this gene interacts with vitamin C, so that someone who has the variant might need a higher level of vitamin C. And then they give some food sources. So the next page on this report gives this person's results. So here it shows for whoever sent it in. Again, it summarizes or recaps what gene they're looking at, what marker, whether they're looking for an insertion or deletion, what the risk variant is, meaning that's the allele of the gene that's going to cause potentially an increased need for vitamin C, and your variant. So this person has the insertion rather than deletion. So then they're saying for this person, their risk is typical, meaning that, sorry, I can't get this thing on the bottom of my screen to go away, part of the PDF anyway. Their risk is typical, meaning there's not an altered requirement for vitamin C. So in this example, what I want you to see and be able to interpret, to know that this is the gene they're looking at. They're looking at one insertion or deletion. This does not specify exactly where on the gene that insertion or deletion is. Remember, the gene's going to be a lot of bases long, of base pairs long, and so I don't know, based on this report, I wouldn't be able to tell someone exactly where the insertion or deletion, or exactly what that insertion or deletion was. So that would be something you would have to look at a little bit further if you wanted to be able to give that more information. Oops, excuse me, sorry, popped to the wrong thing. I was trying to get back. Here we go. I wanted to get back to this this page, because on this report, a nice thing is at the bottom, you'll notice on these that they list a citation, which is fantastic, because it's a great way to be able to start to pull that research and actually go back to the primary literature that they're using to pull these results. And I think it's critically important that you start to be able to read these types of research articles, publications, and see how that translates. And it's often very interesting to take one of those, read it, and then see how they pulled the information to put it in really consumer-driven information in this report. So these are just some great examples of where you can pull those references. Now keep in mind that you would need more than one study in this area, most likely, unless it was an extremely large and conclusive study, to make really strong conclusions. They've typically given one resource. They probably have others as well, but those are kind of questions to ask yourself. What other research supports this? And then sometimes we have to say, well, there isn't a lot else. Does it mean it's not okay to make this recommendation? Not necessarily, but you need to really weigh that out in your own professional judgment, how strong the research is, and then be able to communicate that to clients or the public as well. Okay, so let's look at another one that gives an example, not with an insertion or deletion, but with a SNP. So let me pull one up. I'm going to skip through MTHFR. This is actually an extremely common one, and so you may have seen that already, so I'm going to pick a different one. So the one I've skipped to here is the ACE gene. And so the ACE gene acts in the body, and it says right up here, directs the body to produce angiotensin converting enzyme. So if you remember back from physiology, that helps regulate blood pressure and in response to sodium intake. So this genetic variant can have an interaction with sodium and therefore affecting blood pressure. So you see their explanation here that those who have the GA or AA variant are at greater risk of experiencing high blood pressure when they eat large amounts of sodium compared to the GG allele of that gene. So again, remember that we have 2 copies of each gene. So the options here are that at that specific SNP, so that one nucleotide that's being looked at, they could either have 2 copies of G, 2 copies of A, or a copy of G and a copy of A. So that's what they're looking at here. So then when we go to the next page in this report, how they use it and give this person's individual recommendations, we again have the reminder that it's the ACE gene. The marker is something we haven't looked at yet, but is really important. So this RS stands for ref sequence, and essentially this is something that is becoming really helpful in the literature for identifying a very specific place on a gene. So because genes are so long, we want to know exactly where on the gene we're talking about. And so when you're reading literature, you will, not older literature in genetics, but the newer literature typically has this included, which makes it really helpful to compare, whether one study is talking about the exact same place as the other. It also helps because, as I mentioned, the gene and the protein often have the same name as this one does. The ACE gene or the ACE protein is the same, and so sometimes that gets confusing. So this RS is really helpful to have. They have identified the risk variant as GA or AA. So essentially the best thing to have, or the lowest possibility of having negative interaction with sodium as related to blood pressure, is if you have the GG genotype, which would be homozygous for the wild type. We could also say it that way. In this case, if we're either homozygous for the variant or we're heterozygous, that puts us at elevated risk of sodium affecting blood pressure. So this person's variant is AA, so they do have an increased risk and should limit their sodium. This is one of those examples that it's helpful to look or to consider that this is an issue that their risk is elevated. Their risk for high blood pressure is elevated only when sodium intake is high. So it's that, again, thinking about kind of terminology with nutritional genomics, it's interaction of sodium with their genes based on their genotype to affect a health outcome such as blood pressure. Now we know there's not only one single thing that impacts blood pressure, so it doesn't mean this would be, the end all be all in recommendations related to blood pressure, but it certainly could be a piece of the picture. And it can also help us think why some people can eat canned soup and bacon and everything else that we would consider just horrific in sodium content and have no blood pressure problems, where someone else really is very sensitive to that. Some of these genetic and genomic types of things really help us start to explain that. So as I kind of wrap up, I'm just going to jump back here just a minute. So just jumping back to the PowerPoint, kind of thinking about interpreting these nutritional genomic tests. The very brief example I showed you is something that I'd like you to spend some time looking through those types of tests. Google for a search or pull up the website here for Nutrigenomics and look at their example. Again, it was just one that had a really good one that was easy to find for a sample test, but there are other companies that do testing. Genova Diagnostics has some nice tests. 23andMe has, I am not sure how many sample results are on their website. So any of them, because really those, particularly those direct-to-consumer testing kits, I think it's important to be able to help someone understand what that means and help you start to sort of tease out what's in those reports and what does that mean, and does or doesn't the research support it. Again, I'm not promoting or not promoting the specific company that we just looked at their report, but I think it always is on your shoulders to not only take what they've put together for consumer information, but go back to the original literature and do some of your own analysis as well, because that's what separates you as a professional and as educated in this area from a general consumer. So really important to start to read that literature. That's a huge thing that I want you to get from this week's unit, is some of the terminology and background, so that when you read the literature, it makes a little bit more sense what they're talking about, or the questions, the areas that you need to question or really refer to. So hopefully this gave you a little bit of a orientation, basic overview of nutritional genomics, again, with the goal that you can move forward in reading a little bit more in those areas and have it make a little bit more sense for you.